Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. Yes, welcome to my session. So yes, please do like, share and subscribe. So Rang Bahadur Yadav, yes, hello, hi and welcome to my session. So please do like, share and subscribe. Hi Prince, hi Keshav, yes, thank you so much both of you for joining my lecture. So please do like, share and subscribe. So today we are in lecture 4 of Mendelian Genetics. So we have a lot of questions that I've got today. So based on your inheritance of 2 genes, so we will solve the questions and then we move forward with your chromosomal and linkage. Alright, yes. Yes, Prince. Hello, hello. Okay, so all of you have done the homework questions. So I gave you 7 or 8 questions as homework. So are you done with your homework questions? So shall we solve the homework? Yes. So without any further delay, we will move in because I have a lot of questions today. So let's see how much we can finish. If you're not able to finish, then that will be your homework. Okay? Yes. Okay, so one more announcement I have. So today once again at 6 p.m. we are meeting and we are starting the chapter biomolecules. So this is lecture 1 of biomolecules. So this is at 6 p.m. Okay, so this is biomolecules. So please join me at 6 p.m. for this lecture of biomolecules. And before that, please do share the link with your friends. Please do like, share and subscribe. Alright. Yes, so lecture 4 of principles of inheritance. So we have been talking about Mendelian genetics and we have been solving sums based on inheritance of 1 gene and inheritance of 2 genes. So today, so actually we have discussed all this. So based on this we are solving questions. So based on inheritance of 1 genes, Mendel proposed two laws, and based on inheritance of 2 genes, he proposed 1 law, that is law of independent assortment. Okay. Okay, so we haven't reached recombination and linkage. So I will tell you once we go. So once we solve the questions, we are going to talk about recombination and linkage. So that time I will tell you. Okay, Keshav? Yes? All set? So have you solved the homework questions? Yes, can I move forward? Yes, yes Vishal, a very happy good evening. So thank you for joining my lecture. So hi Satyendra, yes, a very happy good evening. Thank you for joining my lecture. Hi Shimpi, yes, hello, very happy good evening. I'm fine, thank you. How are you? Alright, so everyone is here on time. Okay, so these are the topics that we were discussing. So I hope you remember inheritance of 1 gene, inheritance of 2 genes in detail. Today I'm going to teach you something very different apart from that, and then I will go to homework question because this is something important. So based on this also I have question. What is this term known as epistasis? Epistasis. So I have a gene here. I have a gene here. Okay, so this gene has alleles. This gene has alleles. So please pay attention as to what is epistasis. So when 1 gene at a different loci masks the expression or inhibits the expression of another gene at a distant loci, so such a type of interaction is your epistasis. Suppose if this gene masks the genotype or phenotype of this gene, then this is known as epistatic and this is known as hydrostatic. Okay, so epistasis happens between 2 different genes. So we call this type of gene interaction as intergenic gene interaction. Yes, clear? Yes, epistasis is a type of gene interaction. Okay, so this is epistasis. So what is dominance? Law of dominance we have read, right? Yes, so dominance is intragenic. So dominance happens between the alleles of a same gene. So between 2 alleles of a same gene. So dominance is intragenic, or you can call it as intra-allelic. So this is the difference between epistasis and dominance. So please make a note of it. Yes, quick. So the epistatic ratio, so epistasis experiment was performed in certain plants, and epistatic ratio is 12:3:1. So the ratios are important. 12:3:1. The functional interaction between genes, okay, so you can put it anywhere. Okay, so this is epistatic ratio. Okay, the next one is complementary genes. So I hope you're making a note of it. So this is not there in your textbook. Yes. So the next one is complementary genes. So complementary genes are present in your sweet pea. So Lathyrus orodatus, so that is sweet pea, and the ratio is 9:7. I will tell you what is complementary gene. So Keshav says, ma'am, explain once more. So epistasis is a type of gene interaction, and interaction happens between two different genes at two different distant locations. For example, this is one gene with one set of alleles at a different location. So this is inhibiting or masking the expression of this gene which is present at a much distant location. So this is known as epistasis. So since this is being dominant over this gene, this is known as epistatic, and this is known as hydrostatic. The ratio is 12:3:1, and since it happens between two different genes, it is intergenic. Okay, so law of dominance, so you know what is dominance from inheritance of one gene, right? So this is intragenic. So this is between the same gene or within the same gene but between two alleles. So this is interallelic. Okay. Yes, so distant loci or locus, so locus is a specific position, so this is singular. So when I have more than gene, so it is loci. Okay, Vishal? Yes? So this gene's expression is getting masked, right? So this is masking this gene's expression. The gene which is not getting expressed, that is hypostatic. The gene that is getting expressed is epistatic. Alright, Keshav, did you understand? Yes? Yes, Shubham sir. Hello, very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Yes, everyone understood what is epistasis? So Shimpi, did you understand? Satyendar? So the epistatic ratio is 12:3:1. Okay, so now I move forward to what are complementary genes. So complementary gene interaction is seen in your sweet pea. The ratio is 9:7. So you have two gene pairs. They interact to produce a particular phenotype. Okay, but if suppose I have a gene A, B, B small b. The dominant allele of this gene is not expressed until the dominant allele of this gene is expressed. So this is complementing each other. So you will have A and B expressed only if these two are present together. Otherwise, it will not be present. Okay. Epistasis in squash fruit. Yes, you can say. Yes, that is also right. So a hypostatic gene. Okay, so in epistatic interaction, so this gene masks the expression of this gene. Okay, so this is getting recessive. This is not getting expressed. So this is known as hydrostatic gene. Okay, Jyoti? Yes? Hypostasis or okay, so it is not hydrostatic. Okay, I have written wrong. So that is why confusion. Okay, epistatic, hypostatic. All right, yes, fine. So no, don't confuse with the example. So if I start giving you example, then it will go much detail. So remember, there are two gene pairs. They interact to produce a different phenotype, okay, or a specific phenotype, and the dominant of this character is not expressed unless the dominance of this gene is expressed. So for this A to be expressed, here also dominant character should be present. So this is your complementary genes. All right, yes. So the ratio is 9:7. All right, clear? So wait, I have not started pleiotropic genes, Jyoti. Okay. So I have one more type of gene that is known as supplementary genes. Again, I have two pairs of genes, okay. I have two genes which control the phenotype of a trait. Here I have two genes which control the phenotype of a trait. But so out of these two genes, in one gene the dominant character is expressed only if the dominant character of the other gene is expressed. Okay, whereas the dominant character of the other gene is expressed irrespective of this dominant gene. So pay attention, I am repeating, supplementary genes. Okay, so in supplementary genes I have two genes which control a particular phenotype. Okay, so out of these two genes, the dominant feature of one gene is present only if the dominant gene of the other gene is present. Dominant allele of the other gene is present. Okay, but whereas for the other gene, its dominant allele is present even if the dominant allele of the other gene pair is absent. So this is supplementary. So one needs both the presence, the other does not need the other's help. So this is supplementary gene interaction. So this is seen in your snapdragon or your dog flower. So this is your anti-hirindum species and the ratio is 9:3:4. Yes, has everyone understood? Yes, 9:7. So this also you can write it as 9:7 sometimes. Okay. Otherwise you can split and write as 9:3:4. All right, Vishal. Yes, is this clear to everyone? So this bit I haven't taught. So before going to chromosome and linkage, so this I am teaching. Non-allelic. Okay. So if I have two genes, one gene for height and the other gene for eye color, right? So there are two different genes and they have two different phenotypic expression. So the alleles are called as non-allelic. They are not homologous, right? So it's non-allelic. Okay, Jyoti, did you understand? So is this clear? So I hope you have solved your homework questions. Yes, quickly give me the answer. Yes, everyone quickly give me the answer. So we stopped here. So question number 8, what is the answer? Yes, Vishal, Keshav, make it quick. What is the answer for question number 8? Yes, quick. A is absolutely right. So everyone has got option A. Yes, ma'am, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. Yes, everyone has got answer A. No doubt. How do you solve? So what proportion of offsprings obtained from the cross? This and this will be completely heterozygous for all genes segregated independently. Okay. So here it is a tri-hybrid cross and the cross is capital A. So what will be completely heterozygous for all genes? So completely heterozygous means resembling one of the parent. So they are asking for the recessive offspring, right? So here the recessive offspring will be 1 by 8. So you will have 8 gametes, right? So this is a trihybrid cross. So it is 1 by 8. So heterozygous for all genes means this condition, meaning it should resemble the recessive parent or the second parent. Only one offspring will be resembling this parent. So it is 1 by 8. Okay? Yes? It's trihybrid. 1, 2, 3. It's trihybrid. Okay, Keshav? By confusion? Huh? Okay, so question 9. Yes, what is the answer for this question? So Vishal had asked me this question, right? You have done such question. Okay, so question number 9. So Prince, are you understanding? Yes, what is the answer for question number 9? Hi Anthony, how are you? So thank you for joining my lecture. Yes, a very happy good evening. So please do like, share and subscribe. Yes, I texted you back, Vishal. Yes, how do you solve this? What is the answer to this question? Yes, what is the answer? B. Shimpi, absolutely right. Yes, Prince, you are also right. It is option B, a white flowered mirabilis plant. So it is red flowered and it is white flowered. So your mirabilis plant shows incomplete dominance, right? So these are the parents. So the gametes will be R and R. So this is your F1 offspring. So they are asking in F2 offspring, right? So into capital R, small r. So I do the crossing. So it's like this, right? So the genotypic and the phenotypic ratio is 1:2:1. Total plants are 120. Okay, so how do I divide? So this is 30, this is 60, 30 and 30. So this is how I divide it equally, right? So which means I have 30 white plants and 90 of 2 different varieties. One is red and one is pink. So it is 30 white, so this is right, and 90 of 2 different colors, right? 60 plus 30 is 90. This is non-uniformly colored because it has red, it has pink as well. So the option is B, 90 non-uniformly colored and 30 white. So this is option B. Now you understand? Yes? Yes. So tomorrow we are starting the first previous year question. So please be there. Okay? Yes? Yes. So this is option B. I hope everyone 30 red is your answer. Okay, as long as you have understood this question, I am fine, Keshav. Okay, so the last okay, not the last question. Yes, question 10, what is the answer? Come on guys, quick. So question number 10, what is the answer? Quick, you should only take not more than 2 minutes to solve questions of these types. Once again, it is mirabilis plant. So mirabilis plant shows incomplete dominance. Right? So when okay, so Vishal says, Keshav says yes. So it's very clear because your phenotypic and your genotypic ratio of your incomplete dominance is 1:2:1. So homozygous red flowers are crossed with homozygous white flowers, and in F1 progeny you have pink flowers, and if you do selfing, they are asking you the ratio. So that is 1:2:1. So this is an easy one, and then I gave some homework questions. So I gave 5 homework questions. To that I have added 10 more confusing questions. Yes, tell me question 1. The types of gametes formed by the genotype RY, capital R, small r, capital Y, small y, R. Yes, quick. Quick. Yes, lonely loser. Yes, hello, hi. After a very long time you are here. How are you? Yes, thank you so much for joining my session. So please do like, share and subscribe. Yes, option A. So it's easy one. So they are asking for the genotype. So you have to find the gametes. So this is capital R into capital Y, capital R into small y, then small r into capital Y, small r into small y. So it is directly finding the gametes, right? Okay, so question 2. In law of independent assortment, how many factors are involved for a dihybrid cross? Yes. Question number 2 of your homework. Yes, what is the answer? In law of independent assortment, how many factors are involved for a dihybrid cross? Yes, Prince, what is your answer? Yes. 4. How many factors are involved? Why 4 factors are involved? What are factors? Factors are nothing but genes. So in dihybrid, how many genes are involved? In your dihybrid, how many genes are involved? Yes, 2 genes. Yes, this is the right answer. Why confusion? Okay, lonely loser, you're fine. Okay, that's nice. So here it is option A. Okay, yes, lonely loser, right. So question number 3. Yes, what is the answer? Question number 3, what is the answer? In Mendel's experiment with garden pea, round seed shapes was dominant over wrinkled seeds and yellow color was dominant over green color. So it is simply like this. So you have round shape, yellow color and round shape. So it is dominant over green color and wrinkled seeds. Right? What are the expected phenotypes in the F1 generation of this cross? Okay, so they have taken first R, I have taken first Y. That's all. It will not change anything. Okay, so they are asking in F1 generation. So here this will be the gamete. So here this will be the gamete. So this is F1 generation, right? So I don't have to do. So this is yellow color plus it is round shaped, right? So only round seeds with yellow cotyledons. So this is absolutely right. YD. YD. They're asking F1 generation, right? So it is a simple cross. You have to stop here. Why are you going further? Yes, I remember super fast NEET, but he is not coming these days. Yes, so answer is A. Right, so you know, now you understood how did we derive at answer A? Yes, is it clear? Okay, so question 4. So in a cross between pure breeding pea plants having round yellow-green wrinkled seeds, find out the total seeds having yellow color in the F2 generation, having yellow color in the first generation. It's an easy question. Yes. Yes, what is the answer? You do not have to do the cross. You can do it quickly. It's a dihybrid cross, right? Yes, what is the answer? B, 10. No, B is not the answer, Shimpi. Yes, quick. Yes, come on. No, Keshav, B is wrong. C is also wrong. Yes, try. Only 2 more options are there. Okay. So, looking at the question, it's clear they are asking about dihybrid cross. Right? So, this is the first thing you have to note. If it's dihybrid, monohybrid or trihybrid, right? Yes. The next thing, they are asking you to find the total number of seeds having yellow color. So this talks about the phenotypic ratio. So this is the next thing that you have to identify. In your dihybrid cross, what is the phenotypic ratio? 9:3:1. 3:3:1, right? Yes. So here it is yellow round, here it is yellow wrinkled, here it is green round, here it is green wrinkle. This is the order, right? So they are asking number of yellow color. So 9 plus 3 is 12. So here your right answer is 12. So Shimpi gave the right try. Yes, Prince, absolutely right. Vishal also right. So here it is option A, that is 12. Yes, great. So this is how you have to find. So this should not take minutes. So it should be easy to solve. Okay, so please don't make mistake. Yes, question 5. So in a cross between pure breeding pre-plants having yellow round and green wrinkle seeds, what will be the ratio of seeds having yellow and green seed color? Yes, it's a nice question. Yellow and green seed. Yes, what is the answer? I'm getting variety of answers. Why so? Yes? Okay, so Keshav's answer is right. So it is 3:1. Why, Keshav? Give me the answer. Give me the explanation. So what is 12 by 4? What is 12 by 4? No, it's not C, it is B. Why? Why it is option B? So in a cross between pure breeding pre-plants having yellow round and green wrinkle seeds, what will be the ratio between seeds having yellow and green seed color? So here, so we are talking about color. So this is phenotype, right? So this is yellow round, this is yellow wrinkled, green round, green wrinkled, right? So it is 12:4. Right, so these two are yellow, so 9 plus 3 is 12, these two are green, 3 plus 1 is 4. So this is nothing but 3:1. Right, so it is 3:1. So this is how you get, this is how you arrive at the answer. So the right answer is option B. Yes, wasn't it a very interesting question? In 1 second they confused you, right? Okay, so question number 6. So if dwarf plant was treated with gibberellic acid, so recently we saw the application of your gibberellic acid. It grew as tall as a pure tall pea plant. If this treated plant is crossed with a pure tall plant, then the phenotypic ratio of it is likely to be. Yes. Yes, give me the answer. Yes, such a lovely question. Okay, option C. Why? What does gibberellic acid do? It causes elongation of the stem, right? So your dwarf plant, when it was treated with gibberellic acid, it became a pure tall plant, meaning it has become like this. So now when you cross it with a tall plant, so everyone will be tall, right? So the answer is all tall. So option D is the right answer. Yes, Vishal, absolutely right. Okay, so question number 7. In sweet pea plants, the presence of dominant C and P genes is essential for the development of purple color. So the ratio of plants producing flowers of different colors in the progeny of the cross will be. Come on, please think. Yes, Vishal, you're absolutely right. So that is the role of gibberellins. But Anthony, so here no abscission will not play a role. So here it says that it can grow in length. So that plays an important role. Yes, what is the answer? So please read the question. Take your time. Yes. I'm solving so much of questions so that you don't lose confidence in this chapter. It is not such a tough chapter. D, 3 purple and 5 white color plants. Yes, absolutely right, Vishal. So D is the right answer. Good. Really? Are you telling this as a joke? Are you telling this for genuinely? Keshav? Yes? Okay, so there's someone, future medico. Yes, hello, hi. So you don't know genetics at all. So recently I have taught genetics. So please go back, watch my lecture. So if you are able to understand, fine. If you don't, ask me again. Alright, so anyways, thank you for joining a session. So please do like, share and subscribe. Okay, so how did he get this? Capital C should be there, capital P should be there, only then you get purple flower. Okay, the cross is this. So from this, what is the gamete? Capital C, capital P, capital C, small p, small c, capital P, small c, small p. So from this, I took out the gametes. So here the gamete will be capital C, small p, small c, small p. Right? So now do the crossing. So now do the crossing. C, C, P. The very reason for I changed the color is so that you understand. Oh my God. So this is capital C, small p, small c, small p. Okay, I hope you understand. Okay, so here then you have capital P, small p. Oh, what is happening to this board? This is the cross, okay? Yes, I'm a good teacher. Okay, thank you so much. Yes. Okay, thank you so much, Keshav and Vishal. So, thank you. Yes. So, how do I find the gametes for this? So, it is C, capital P. So, it is C, small p. So, it's C, P, C, small p. So, here it is capital C, small p, small c, small p. So, do the cross. So, wherever there is C and P, it is purple flower. Other than that, it is white flower. So, find out how many purple flowers are there. So, this is double C. So, C is there, P is also there. So, this is purple flower. This is purple flower. Okay, and then you have C and P. So, this is also purple flower. So, the remaining has only dominant C or dominant P, right? So, 1, 2, 3, 4, 5. So, you have 5 white and 3 purple, right? So, the option is D. Clear? So, Shimpi, did you understand? Yes? Okay. So, it is 3 purple and 5 white colored flowers. Yes, question number 8. So, Mendel crossed a pure white flowered recessive pea plant with a dominant pure red flowered plant. The first generation of the hybrids from the cross would show. Yes, question number 8. So this is Mendel and he did it in your pea plant. So don't just go white and red, so it is incomplete dominance, it is not incomplete dominance. Yes, quick. Yes, what is the answer? Yes, absolutely right, Prince. So very good. I am really happy. Good, Keshav. So this is Mendel's cross, right? So white flowered is recessive, red is dominant, white is recessive. So in the F1 generation, it will be like this. So this is pea plant. So pea plant does not show incomplete dominance. So all are red flowered plants. Yes, absolutely right. Okay. Yes, Keshav, good. So question 9. So from a cross of this following genotypic expression obtained in the F1 generation will be. Come on, please take your time and give me the right answer. Quick. Yes, make it quick. Yes. C. No. Shimpi, no. Prince, no. So the cross is this. So here the gamete is capital A, capital B, small a, capital B. So here there is only one gamete, small a, capital B, right? So now how do I do the cross? So one of this, one of this. Option A. Why B, why C, why D? Yes, it's a direct cross, right? In the F1 generation, then why so much of confusion? Yes. Why so much of confusion still? For everyone? Okay, so moving forward, so question 10. A dihybrid for qualitative trait is crossed with a homozygous recessive individual of its type. The phenotypic ratio is. So it is crossed with a homozygous recessive, meaning it is your test cross. Now give me the answer. So I should come up with a rapid fire test for genetics, especially these crosses, right? Only 25 seconds for each question. That's okay. You took time and you are gaining confidence by doing it on your own. Yes, that's good. It's not bad. Yes, should I come up with rapid fire questions for Menti? Yes. So I will bring quickly, very quickly, no leaderboard, directly the answer. Huh? Yes? Answer is D. No, Antony's answer is right. Antony and Prince are right. It is 1:1:1. How? So a dihybrid. So in your dihybrid, so this was the case, right? It is crossed with a homozygous recessive, meaning the F1 generation is crossed with your homozygous recessive. So for here, the gametes are so here only one type of gamete. So do the crossing. So each one is unique, right? So 1, 1, 1, 1. So 1:1:1. So option is C. Yes, it's a test cross. Absolutely right. Okay, so here it is option C. So Keshav is getting one question. He is not able to solve the next question. Alright, so question 11. The percentage of heterozygous individuals obtained in the F2 generation from selfing the plants with genotype capital R, small r would be. Yes, quick. Yes, the last few questions. So with this, we'll be done with inheritance of one gene and two genes. So in the next class, I will move to chromosomal chromosomal theory, linkage, mapping, all that. So there also we will solve questions. Then we have genetic disorders. There also we will solve n number of questions. 75, yes, okay, 75 is the wrong answer. Shimpi, 75 is the wrong answer. Yes. So percentage of heterozygous individuals obtained in the F2 generation from selfing of the plants with capital R, small r. So this is your monohybrid cross. Right? So in your monohybrid cross, it is like this, right? Yes, so together 4 makes 100. So contribution of one offspring is 25. So 2 offspring is heterozygous. So that is the question. So it is 50. So this contributes 25%, 50%, 25%. So the answer is 50%. The percentage of heterozygous individuals obtained in F2 generation from selfing the plants with genotype. So this is the only genotype, meaning it is monohybrid. If it is not mentioned mirabilis, dog flower, etc. So it is normal Mendelian cross. Okay, so that is how you should consider. So the answer is option B, that is 50. Okay, so question 12. In a dihybrid cross, 80 plants are obtained. Okay, so the ratio of homozygous and heterozygous shall be. The ratio of homozygous and heterozygous shall be. Yes, quick. It's an easy question, provided you have to think. Please think and give me the right answer. Come on. Please think. Yes, Shimpi, absolutely right. Perfect. Fantastic. Yes, Vishal, good. How? So, in a dihybrid cross, totally you have 16 offsprings. Right? So, it is given 80 plants are obtained. So, 80 by 16 is 5 plants of each type. Right? So, the ratio, so it will be 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. So, this is the ratio. Of this is homozygous dominant, this is homozygous recessive. So, each one contributes 5%. Right? So, this is 5%, this is 5%. So, it is 10 is to remaining, that is 70. They want the ratio of all the homozygous dominant as well as homozygous recessive. So, it is option B, that is 10 is to 70. Okay, clear? Yes, moving to question number 13. So, in F2 dihybrid ratio, 9 is to 3 is to 4 is explained on the basis of. Yes, now only I told you. So, this is what type of interaction? 9 is to 3 is to 4. Yes. So, epistatic is 12 is to 3 is to 1, right? Supplementary is 9 is to 3 is to 4. So, allelic interaction is your dominant interaction. So, that is your 3 is to 1. Okay. So, here complementary gene interaction is 9 is to 7. So, your right answer is supplementary gene interaction. Yes, Prince, absolutely right. So, very good. Thank you. Yes, question 14. A self-fertilizing, meaning selfing of a trihybrid plant forms. Selfing of a trihybrid plant. So, selfing of a trihybrid plant forms. You gave so many wrong answers, but still it is your favorite subject. Yes, one day you will become very strong. Don't worry. Yes, how many gametes you will have and how many zygotes will you have? You have 32 different zygotes. Where did 32 come from? Yes, you're right, Prince. So it is 64 zygotes. So in your dihybrid cross, so it's 2 power 2, you have 4 gametes and the offsprings are 16, right? So this is 4 square, right? So here it is 2 power 3, that is 8. So 8 square, it's 64. Okay, so it is 64 different gametes. So this is the formula for this type of question. So Vishal, did you understand? Yes. So let's move on to the last question for the day. So question number 15. So the phenomenon in which an allele of one gene suppresses the activity of an allele of another gene is known as dominance, inactivation, epistasis, suppression. Yes. Yes, Vishal, right, it is epistasis. I guess Vishal gave answer for the previous question. Okay, yes, no, it is not dominance. So, one gene suppresses the activity, allele of one gene. In dominance, one allele suppresses the another allele. So, in dominance, it is within the same gene. So, here you have one gene and another gene. So, it is epistatic interaction. Alright, yes, absolutely right. So, with this, I am done completely talking about inheritance of one gene and inheritance of 2 gene. Okay, so next we'll have to go with your chromosome and your linkage. Actually, I got the slides, but it's okay. We'll start in the next class because in this also we have a lot of problems which we will solve. So I will meet you with this chapter. So today is Wednesday. I will meet you on Friday where we will solve a lot of questions once again. But please do join me this evening at 6 p.m. for lecture 1 of biomolecules. So I will try to finish biomolecules as quickly as possible. I will not drag on. But in biomolecules also I will bring a lot of questions because in biomolecules the questions can be tricky. Alright. Yes, Anthony, what is your doubt, please? Yes, ask me. Yes, thank you, Prince. So you enjoyed the question. Okay, so you got an idea how to handle this type of question. Yes, that is what I want. So, okay, Keshav, great. So, yes, Anthony has some doubts. So, I will solve and then I will leave. But please do join me at 6 p.m. Alright, yes. So, thank you everyone for joining my lecture. So, if anyone comes to you and says genetic is tough, tell them no, we have solved so many questions. Okay, yes. So, please don't make any mistake. Even in the middle of your sleep, you should solve it properly. Okay, yes. Yes, Anthony, what is your doubt? Type in for us. We are waiting for you. Yes. So, tomorrow we are having our first session of PYQ question. So, I am going to solve 2020 question paper, only class 11 questions. So, the questions that have come from class 11 chapters, only that I am going to solve in tomorrow's lecture. So, tomorrow PYQ session is there at 6 p.m. Alright, okay. Amino acid derivative is epinephrine. Yes. So, epinephrine, it's a precursor, is an amino acid derivative. Alright. Yes. Today, it's not okay. Tomorrow, 6:30, we don't have a session. 6 to 7, we have a PYQ session. Instead, Saturday, we have a Menti. Alright, Vishal? Yes. So, Anthony, clear? Okay, so I will meet you guys at 6 p.m. this evening talking about biomolecules. So until then, take care. So this is me wishing you most and more, and this is me signing off. So bye bye and take care.